so you can read it, Meg. Sorry. I was just telling Meg that I'll just scooch over so she can actually read what the screen says. And I'll kind of gauge where everyone's at once I see how writing is going. Everyone has the general idea of what I want from there. And now, if you could draw what you think a neuron looks like, and we'll go from there. And again, this is just your initial model, so if you can make it nice and big, that would be fantastic as well. That's why I wanted to give you guys tons of space, so. I'll give everyone like 30 more seconds and then I'll start going through stuff. So now we're gonna do a little bit of a show and tell and a little bit of share so I can kind of gauge where everyone's at. Why don't we start with Evan? Okay, Evan, so what do you know about the nervous system? What do you know about neurons? And what do you know about how neurons connect?
The nervous system is one of the systems of the human body.
Yep.
I think it spread throughout the body with nerves. There is like, I don't know how to say it, there's like a direct one where it kind of like moves up, like signals from some body part moves to the brain, and then like you can act on it or whatever. And then there's some that like is like pain, where like it doesn't travel all the way to your brain, but your body just kind of knows it's not good for you, so like you stop doing it.
Yeah. Can you hold up your model so I can
yes,
see?
sure. This is what I drew.
Amazing.
As like neuron. I know it has like some round part and like some kind of tail looking part.
Yeah.
Maybe it is supposed to send electrical signals because I think that's how they communicate with things.
Yeah.
That's about it.
Awesome. Thank you for sharing. Okay, we'll do Darcy next.
I totally forgot to talk about like the autonomic nervous system. Nervous system.
Oh, that's fine, don't worry.
I like just completely missed the beginning steps. I talked about like parasympathetic and sympathetic,
Yep.
where the parasympathetic is the rest and digest, the sympathetic is the fight or flight. I always want to mix them up because I think of paracupas and I think of the flying, but I'm like, no, it's the opposite. I also put like dendrites and axons, myelinated sheath, neuroramvier, action potential, acetylcholine.
Yeah, and can you show
I
us
didn't
your
draw
model as
that
well?
big, I just drew a little
Yeah.
dendrites,
Yeah.
the myelinated sheath, and the axon.
Yeah. Awesome. Thank you for sharing. And how about you, Meg?
I think the nervous system has something to do with the spine, and it's connected to the brain, and neurons look something like this, and they use their like tentacles to talk to each other.
Yeah, that's awesome. And so, Anson, the only thing you missed was I basically just posed these questions. What do you know about the human nervous system? What do you know about neurons? And what do you know about how they connect? And I just got everyone to basically just draw their like initial model. So if you just want to draw like what you think a neuron looks like initially, and then I'll go from there.
Sure.
Yeah. I am so excited for you guys to learn about neurons today. It's like I am such a nerd with this stuff, and this is exactly what I love. So. And I unfortunately don't get to teach any of it on law practicum, so it's kind of a disappointment, but no. Awesome. Okay. So I'll go to the next one. So you guys can flip over to the next page. So it's just on the back side, and this is kind of your intermediate model. So this is where I'll kind of be giving you some more information, and hopefully it'll help revise your later sketch of it. So, a neuron, this is what a neuron looks like. So you can see there's a cell body, or like a specific section of it where the nucleus is housed. So the nucleus is indicated by this little purple circle in the middle of it. And then the cell body, or how it's referred to in people who study the nervous system, it's called the soma more specifically. So that's sort of what this is. And your final model, I am hoping that there will also be labels on your stuff, and there is also a bit of a worksheet after that I'll get you guys to go through. So if you want to add labels to like feel free to redraw it now, and then you can add labels to it, and then just like write down certain information that you think is important. On top of that, there's also these kind of more extensions that come off of the soma. There's two different types. One is the axon, and generally neurons just have one axon. And then the little scraggly ones that you see coming off of here, that there's like a bunch of small ones that kind of branch off. These are called dendrites. So the reason that you have so many dendrites versus just one axon is the neuron itself wants to kind of create more surface area where it can kind of take in more information, right? So if you think about it, like in your skin, you have neurons all over your skin, and those are more for like sensation or tactileness is what it's called. Think of it if you have a neuron that only has one dendrite. What would the point of that be if it was in your skin? Because if you think about it, you have so much feeling all over, right? Like you can feel, so everyone, if you want to hold up your hand, when you're touching your hand, you can feel so much sensation, right? If your neurons only had one single dendrite, you would not have such a strong kind of sense of kind of tactileness, and that's the same for all of your senses, right? So that's why you have so many dendrites that kind of branch off from here. This spot at the base of the soma, so at the very bottom, this is where all of that kind of electrical information and all the like stimulation gathers, and this is called the axon hillock. So I'm not necessarily going to get into action potentials today, just because they're kind of their own thing, and I wanted to keep it more kind of straightforward with just general structure and general function of neurons. But this is basically where an action potential would be initiated, is at the axon hillock. And then this really long kind of like outbranch of the neuron is called, again, the axon. So the dendrites, think of it as there's lots of them generally, and they collect information for the neuron to form an action potential or an electrical signal. And the axon is where this signal gets sent along it. So it kind of gets brought outward, and it will end up at the far part here, called the axon terminal or terminal. So generally there's just one long axon, and it's generally just a couple terminals, but it can sometimes be more kind of spread out like this one is here. And there's also this blue bit here, which Darcy had touched on before, and this is called the myelin sheath. So the myelin sheath is like an insulated layer, basically. So this is just to help with that propagation of that signal. So it actually helps increase it if there is myelin present on the neuron. And these little pieces of unmyelinated bits on that axon are called the nodes of Ranvier, and they're basically just the exposed bit of the axon where there is no myelin sheath on it. Does that make sense to everyone? Yes?
Why is there no myelin sheath on all of it?
I'm actually about to get into that right
I
now.
also have another question.
Yes, yeah.
Is neuron a cell?
So the neuron is a cell, yeah. Yeah. It's like the specialized cell for the nervous system, basically. And there's actually two special types of cells for the nervous system. I'm not necessarily going to get into the one because it's just super complicated and just overly complex, and you don't learn it in high school, but you learn it at a much higher level. Okay, so I'm going to play this video. I want you guys to kind of keep keep things in mind when I'm showing it. The first one we'll talk about the size of the axon, so the diameter of it. Pay attention to whether a larger axon propagates a signal quicker, or if a smaller axon does. So I'll play that. Try to ignore all the big words because I couldn't find anything that wasn't super intense with talking about physics stuff. So if you want to also write that down, you can write that down, saying like, keep in mind size of neuron, how that affects propagation of the signal, and also myelination. So why is this myelin sheath on it, and why are there bits and pieces in between? I have to exit out of this because my embedded video unfortunately did not work. Here we go. So this man is also very dry. I'm so sorry. I tried finding something that was entertaining, but it was not. So, okay.
Understand the second mechanism, we first must discuss where myelin comes from. Myelin is provided in the body by two different specialized glial cells, the Schwann cells and the oligodendrocytes. Schwann cells myelinate individual segments of the axon in the peripheral nervous system, as well as the spinal nerves and the cranial nerves 3 to 12. Oligodendrocytes myelinate multiple axons, sometimes up to 30, in the central nervous system. Oligodendrocytes are also responsible for myelination of the second cranial nerve. To understand what myelin is, we can consider its formation. If we take Schwann cells, for example, and we look at the cross-section of an axon, the formation of myelin sheaths begin when the Schwann cell starts wrapping around the axon. Like any other cell, the Schwann cell has a cytoplasm and a nucleus. Over time, the Schwann cell will continue wrapping around the axon such that its cytoplasm gets displaced out and only concentric layers of membrane hug the axon. This results in the separation of an inner membrane layer named the myelin sheath and the external cytoplasmic layer named the neurilemma. Two very important proteins involved in holding myelin together are the myelin basic protein and the proteolipid protein. Between each myelin segment, you will notice that there are bits of axons that are free. These free segments are known as nodes of Ranvier, and these nodes are very important because they are concentrated with voltage-gated channels. Back to our passive membrane properties, let's consider two axons of the same diameter, but one myelinated and the other not, to see how myelination impacts the conduction velocity. First, remember that as current passively propagates, there is always some loss of current that is partially due to ionic leaks. In myelinated segments, there is essentially no possibility of leakage, and as a result, the current is better maintained. To put it in passive membrane terms, the reduction of channels caused by myelination increases the membrane resistance. Another consequence of myelin comes from the additional thickness it provides to the membrane, which considerably decreases the capacitance by separating the charge at a longer distance and reducing the electric field between the opposing charges. The impact myelin has on the membrane resistance and capacitance can be further analyzed through the time and space constants. The time constant will remain somewhat unchanged because capacitance goes down and resistance goes up. When it comes to the space constant, here again, the increased membrane resistance will yield a higher constant, and as you expect, the conduction.
I'm gonna pause it there because that's a little but yeah, so basically with myelin more specifically, again, actually I'll draw this on the board. So this also ties into, there we go. This also ties into neuron thickness as well. So the one thing that I didn't want to show off that is there's a specific rate of decay that happens with your like signal, right? So the signal will always start really strong, and over time it kind of decreases like that. The thing with a myelinated axon versus an unmyelinated axon, so I'll just draw this like this, is because your signal is only going to be really affected by these little bits here, this decay rate is kind of, how to explain it, there's less decay between the signals, so it kind of propagates it almost does this thing where it jumps from here to here, versus if you have a signal on an unmyelinated axon, it kind of just decreases like this until you have no signal left. So that's kind of the benefit of myelination. A larger neuron versus a smaller neuron, this is something that's also important as well. So basically, if you have like a really thin neuron versus one that's much bigger, it's kind of dealing with how much surface area and how much tension there is, and how much resistance this causes to that specific signal. So a smaller neuron, when you have a signal traveling through it, there's a lot more resistive forces from here because it's so close and so tight knit, versus a larger neuron, you have less kind of influence of those resistive forces because there's more space within the neuron itself. So a larger neuron and a larger axon propagates signals a lot quicker than smaller ones, if that makes sense. Yeah, and so generally in our nervous system, when we have signals that have to kind of pass through our body, so think of it as like the initial pain response. Like you stub your toe and you like immediately feel it, right? Oh, thank you. Think of it as when you stub your toe and you immediately feel it. That's because we have these massive neurons that are myelinated basically from our spinal cord going up into our brain and from our toe going up into our spinal cord. But then has anyone ever had that feeling where you like stub your toe, but then there's like that lingering effect of the pain afterwards? That's because certain pain neurons don't have myelin on it and they're quite small. So that's why you get that lingering effect coming in later on. So that's why you have like that initial pain at first. And then you also have pain that kind of is just lingering afterwards because it's like that slow transmission of that signal. Here, I'll go into this now. So, here we go. So, what information did any of you learn from that small bit? Yes.
Honestly, I had never quite understood why having like the myelinated sheath and the nodes around VA is just like, oh, it's quicker.
Yeah.
Like I definitely knew about the diameter because
Yeah.
we talked about that like in classes with the like giant squid axon.
Yeah.
When we use as an example, but then once they showed the example of the current, I'm like, now it makes sense. Like it honestly was never really explained before.
Yeah, I know. It is quite confusing. Okay, Evan, I'm gonna call out you guys to see any other new information, or just like stuff that you learned even in general, like even about the structure.
Basically about the structure.
Huh?
But I learned like that little ball thing that I had in the beginning was actually like not a ball. I think the most interesting thing I learned a lot, right? Like the updated model reflects that. But I think the most interesting thing I learned was probably about like the fact that there is a lot of dendrites, because they are the one that collect information, and that big area is supposed to be covering all those areas where you want like sense. And there's like one axon that sends it down the chain.
Meg?
I learned like the overall shape of the neuron and like the myelin.
Yeah. And then Anthony, if you have anything new, I know that there wasn't too much of it.
I learned about the idea of leakage and how that affects the speed of transmission regarding the signal.
Yeah. So again, there's so many different things that like we kind of touched on here. Again, there's so many different parts to a neuron that a lot of people generally miss out if you're not too familiar with it. Talking about how larger axons propagate signals a lot quicker compared to smaller
That's
axons.
whatever you just had.
No. This is the oldest computer I've ever used in my life. And then again, axons that are myelinated generally propagate signals a lot faster as well. And to also showcase the effect of myelin, I'm gonna do a little interactive activity. So if I could get all of you, and if you wanna participate too, you can also participate. If I could get all of you to kind of stand, kind of like, I guess in a line across here, and if you can stand like this. So, so like the tips of your fingers touching one another. Beautiful. And so this is to show basically a neuron that is unmyelinated. So just showing the distance that it goes with an unmyelinated neuron. So what you're gonna do, Meg, is I'm gonna hand you the ball, and this will be your signal, and you're just gonna pass it, and then pass it to Darcy, and then you just keep kind of doing that, where you take it and you just keep passing it along. So, go. And then you can count up on the clock how long it takes to get to the end. Okay, so that was around around like 8-ish seconds. So now to show the effects of a myelinated axon, Megan, I'm gonna get you to stand there. Darcy, stay there. I'm gonna get you to stand there, Evan, and then I need you to, Anson, if you wanna go in the middle, and then if you could go at the end there. And what you're gonna do is you're gonna now pass the ball to one another, and we'll see how quickly it can go and how much further you get in the same amount of time. So,
We gotta wait for them to say start.
ready? 3, 2, 1, go. Perfect. See, so that took half the amount of time and you got double the distance, right? So that's just showing you how myelin actually affects signal propagation and how, and also just how it does this thing where it not jumps, but it kind of jumps from section to section. So you all would be representative of the nodes around being, and the space in between you would be representative of myelin. Perfect. Here, I'm gonna take the ball. You all can take a seat now. That was my interactive activity for today. You're welcome. Now you go up here and sit down and relax. Okay. So again, with just the signaling properties in general, again, you have your neuron, you have information that generally will come in from the dendrites. This is not always the case, but it's just a general kind of assumption of it, which then propagates down your axon and goes to the axon terminals. So I'm going to give you guys a little bit of time, maybe like a minute or two, to just write down some more notes for it and to work on your kind of like revised model before I get into the last bit of stuff.
They're just like an updated model, not the finalized model.
Yes, yeah. So just let me update it model. So the final model we'll do in just a second. I'll give you all 30 more seconds and then I'll get started on the last little bit. I'll get everyone to flip to the final model now. Perfect. Okay, so there's I believe two other factors that I want you all to keep in mind as well. So the first one is ion concentration. So this is generally kind of how this signal propagates, is through changes in these ionic, or changes in the concentrations of these two ions, sodium and potassium. I'm gonna go through a little bit of a simulation and I want you all to kind of note where sodium is high versus where it's low, and where potassium is high versus where it's low, because I want that in your final model as well. So just showing like an Na plus with like an arrow pointing up for high, wherever you think it is, if it's outside the cell or inside the cell. And then sodium so Na plus high, whether it's inside or outside as well. So yeah, so that's basically the importance of ions, is that it helps propagate those signals, and I have a little bit of a simulation that will hopefully help kind of understand. So again, just keep note of where the concentration of sodium is, say, high, and where the concentration of potassium is high. And you guys can also come up here and like crowd around the screen if you want, because I know that this is not being projected up on the board, so it can be a little bit harder. And you can also stimulate the neuron itself, so you can kind of see how the properties work and how movement of ions happens as well. So if you hit stimulate, you see the signal comes down, and then you see a lot of movement of different ions, and you can see slight changes in concentration, but it relatively remains the same, right? No. Okay. Does anyone want to tell me where sodium is high? Outside. And where's potassium high? Inside? Yeah. Perfect. Yeah. And this is through FET. I don't know if anyone here has used FET before, first off. They do great simulations and great things. And honestly, like, this is just so fun. I was playing around with this and I was like, this could just be my brain break for the day. It's just constantly stimulating a neuron and just going through it. And you can also see, so this is basically what like an action potential looks like when you're looking at membrane potential versus time. So again, it is fun to just say, oh, I'm gonna stimulate the neuron, and over time you can kind of see where you have that increase in that kind of voltage on the membrane. But yeah, I highly recommend. This is a very fun type of a simulation that you can do just at home. There's a lot of other ones as well that you can look at, but yeah. I do wish there's more manipulation with this, but it's kind of hard to find something that is fun and more interactive like this. That's not just manipulating current and then changing of graphs, because that's a lot of the nervous system, unfortunately. But yeah. Does anyone have any questions about that? Does that make sense? Okay. And then the last thing that I wanted to talk about is connectivity. So, does anyone off the top of their head know any form of connectivity? So how neurons connect between one another? Does anyone have any information on that? There's also no wrong answer. Yes.
Synapses.
Through synapses, yep. Where do those synapses occur? So from what structure to what structure?
It can be between axon and dendrite, axon and a motor plate. Yeah, awesome. So the one that I'm gonna specifically talk about is the axodendritic synapse. So this is basically a synapse that occurs from one neuron's axons onto another neuron's dendrite. So as it says in kind of the title, axodendritic synapse. There's lots of different kinds of them, so I'll draw those on the board as well. So again, axodendritic, and these ones for your final model too, I would like you to draw this one specifically, but you can also, if you wanted to, also write down and look at others, you can. So just my little neuron, so again, this is just very quick. So, dendrites, soma, axon terminals, dendrites of another neuron here, with it going down this way. So that's axodendritic. You can also have axosomatic, and does anyone have any idea about what that might look like? Think of what a soma is. Yes.
Is that big circle?
Yes, yeah. So in the case of axosomatic, what you would get, that's interesting, is instead of the axon synapsing onto the dendrites, you would have it synapsing directly onto the cell body of another neuron in that case. And how about axonic? Where do you think that one would synapse?
Oh, axon.
Yes, exactly. Yeah, so that one, you would have it synapsing onto the other neuron's axon that way. So there's lots of different ways that you can have neurons kind of interacting with one another. This one specifically, the axodendritic, is the most common synapse, and it's one that you find most kind of prevalent in the nervous system. Yeah, and that's kind of why I wanted to highlight it. So I want you all to finalize your model now on that last page, and if you could draw a neuron connecting with another neuron, and then at the very bottom of the page it says, what did you change or what have you added since your initial model? I want you to talk about ion concentration if possible, and I want you to also talk about size of the axon, and also myelination if that affects signal propagation as well, and then we'll go from there. before I go to the end, I'll also get everyone to share their beautiful drawings and share the stuff that they also added on. So yeah, you missed our fun, well, I think it was fun, but our fun little interactive activity showcasing how myelination affects propagation of a signal. So yeah, we can also redo it for you if you want us to, if we have time. So yeah.
And I'll watch it in your video.
Nice. No, and I'll give you guys maybe like another 2 minutes to kind of finish your drawings. They don't have to be perfect. It would just be interesting to see how everyone, like what they change from their initial model. And I think I might get everyone to do that. Showcase your initial model and then showcase your last model and kind of show us maybe what you've learned and some new stuff that you maybe didn't have initially in it. So, no. I'll give everyone another minute and then we'll reconvene and we'll start chatting about stuff. If everyone can kind of wrap up what they're doing, I think we'll reconvene then. Oh, yeah.
You have till 4 p.m. to do it.
Oh, perfect, yeah. Yeah, we'll do a quick discussion and then if you guys wanted to do your peer feedback, you can do that. So let's start with Anson this time. So can you show us your initial model? And then your finalized model now.
So my initial model was just a ball
Yep.
with a squiggly line, a simple squiggly
Yeah,
line,
yeah.
and a few more simple straight lines
Yeah.
to represent the dendrites and the axon.
Mhm.
For my updated model, I included more detail. I kind of made it a better shape. I added the myelin sheath as well as the nodes of Ranvier.
Yep.
As well as include the size of neuron and the myelination effects.
Mhm.
And finally, in my final model, I also included how they attach. They could attach through an axodendritic connection or axosomatic, as well as I also drew, a big channel is faster than a small channel.
Yeah. Was there something that you learned or something that you didn't know was present on a neuron before?
I didn't know that the axons could connect to other places
Yeah.
other than the dendrite.
Yeah.
because
I think that's probably one of the biggest kind of like misconception is that it always has to be axon dendrite and that it could, but like not a lot of people think it could be axonic or axosomatic, right? Okay, we'll go for Meg now.
Are you presenting right now? I gotta check on my groups.
My initial model looks like this. It only has one set of like receptors.
Yep.
And my updated model looks like this. It has all the parts labeled. And the final model looks like this.
Yeah.
And I added how it can be axodendrotic, axosomatic, and axonic,
Yep.
and the concentrations.
Yeah, awesome. Darcy?
My initial was not labeled, but it has, like the cell body, the dendrites,
Yep.
the myelinated sheath around the axon. And then I added labels to a bunch of the stuff. Not much extra was really like drawn. I
That's
think-
fine.
Yeah, I think. And then the final one, the extra was drawn like this, and the axodendritic synapse, and then just a whole whack of labels, and also
Yep.
including like the potassium concentration is higher in the cell and sodium
Yeah, yeah.
outside the cell.
Awesome. And then how about you, Evan?
This was my first model. It was just a ball with a tail on it.
Yep.
We're supposed
to be watching video.
And then my next model now represents that one singular neuron a little bit more in detail. I've labeled most of the parts to it and how it kind of works. And the third, my final model, is basically the same, except now there is a connection to another neuron, which is, I think, what's more important. Because here it was about the singular neuron, now it's about how that neuron connects with another neuron and also how it interacts with the environment in terms of the ion concentration.
Awesome. Yeah. So again, now you all know what a neuron looks like, and I wanted to make something really cursed for the end of it. So you're welcome. And if we had more time, like it is, I think it's time for mixing. But I also had these, so if you guys want to keep these just as like a keepsake, and you can say, look, I know how to label a neuron, and I know functions of stuff now. That's all that matters.
Probably going to copy this.
Yeah.
Actually, although I would not be teaching this in like grade 9 or 10.
No, it is like grade 12, so. Yeah, awesome. Does anyone have any questions about stuff?
Does it, when we drop the ball,
No.
do signals get lost in the neuron?
Signals do get lost in neurons for sure. It is less common in a myelinated axon, but the thing with that is to even form like an action potential, you normally don't lose it in your axons, but you lose incoming signals when they hit the dendrites. So like, say like you have like a neuron, so you have like a neuron that has lots of branches of dendrites, right? And it's kind of like, how to explain it? So this is just my very quick, this to here, and then these are like its dendrites. And say you have like a signal from like another axon and like another neuron here. If it synapses onto this dendrite, sometimes this signal isn't strong enough to make it to the axon helix, so it's just lost because you don't have it propagating enough going through that dendrite. Because there's also a difference where action potential is a active form of movement for the like neuron's signal, but when the signal gets transmitted from an axon to say a dendrite, it's now in its passive form. So it's not necessarily activating like all of those different ion channels, it's just moving along the dendrite. So it gets lost because of its decay rate. So kind of like over time, its like decay rate is like this shape. So it gets lost really quickly unless it's a strong enough signal where its decay rate, even when it gets decayed, it still makes it to that axon hillock. And it just basically summates everything, if that makes sense. Ish. Yeah. No? Does anyone else have any questions? No? Okay. Thanks, guys.